Hello everyone, I'm Yihang Chen. Today I will give a brief talk about the stem cell-based therapy for Parkinson's disease, dopaminergic neuron replacement therapy. First, why do we need to replace dopaminergic neurons? Parkinson's disease occurs due to the gradual loss of dopaminergic neurons in the substantia nigra of the midbrain. This degeneration is associated with multiple factors, including genetic mutations, exposure to environmental toxins, and neural aging. As these neurons die, the dopamine level in the brain decreases significantly, and leading to impaired signal transmission within the basal ganglia circuitry. And as we know, dopamine plays a crucial role in maintaining the balance between excitatory and inhibitory pathways in the brain. So, therefore, when the dopaminergic system is damaged, this balance is disrupted, resulting in motor dysfunction such as tremor, rigidity, and slowness of movements. Next, let's see how stem cell-based replacement therapy works. The goal of this therapy is to restore dopamine production by transplanting new dopaminergic neurons derived from stem cells. Stem cells mainly come from two sources, and embryonic stem cells, which is ESCs, are derived from the inner cell mass of the blastocyst and can differentiate into almost any type of cell, including dopaminergic neurons. And after specific induction factors such as HHH and FGF8, these stem cells can be guided to differentiate into midbrain dopaminergic progenitor cells. And another approach uses induced pluripotent stem cells, iPSCs. This method introduces key transcription factors into adult somatic cells, such as skin fibroblasts, to reprogram them back into the pluripotent states, similar to embryonic stem cells. And iPSCs share similar properties with ESCs and can also be induced to generate dopaminergic progenitors for transplantation. And the use of iPSCs enables personalized treatments and effectively reduces the risk of immune rejection. Well, although both approaches are promising, but challenges remain, including a long differentiation period, unstable efficiency, and the difficulty of obtaining pure and functional dopaminergic neuron populations. And now researchers are continuously improving this process. Once suitable cells are prepared, the next step is transplantation, known as stereotactic injection. Using MRI guidance, surgeons precisely locate the target site, the putamen. And it is worth noting that absolute fixation of the head and sterile environment are necessary. Well, a small hole is made in the skull and a thin injection cannula is slowly inserted along the stereotactic pathway. Suspension containing the dopaminergic progenitor cells is slowly infused into the target area, and the cannula is carefully withdrawn to minimize brain tissue damage. And the transplanted cells will gradually mature into functional dopaminergic neurons, which can release dopamine and help restore the impaired neural signaling. This therapy offers several significant advantages, and first, it provides a long-lasting therapeutic effect as the transplanted cells can survive in the brain and continuously release dopamine. And second, the use of iPSCs allows for personalized treatment and effectively reduce immune rejection. And most importantly, this therapy fundamentally repairs the pathological mechanism of Parkinson's disease, enabling the brain to naturally produce dopamine again, instead of relying on temporary drug supplementation. In summary, stem cell-based replacement therapy offers a novel and promising approach for treatment of Parkinson's disease. Future research will continue to optimize differentiation protocols, achieve standardized large dose production, and enhance transplantation safety to promote the clinical translation and real-world application of this therapy. And this is all the content I'm going to talk about today.